Good morning, everyone. My name is Hilde Eggermond. I'm the chair and the coordinator of Biodiversa Plus, the European Biodiversity Partnership. And it's a pleasure to introduce you to this workshop on capacity building, also on innovative tools and methods for biodiversity monitoring. And the workshop is organized by the colleagues from Denmark, Lars and Toke, so you will get to know them over the next 2 hours. So as a way to introduce you to the workshop, I will just say a few words on Biodiversa Plus itself. I guess many of you may know us, but maybe it's still new for some of you. So Biodiversa Plus is basically the European Biodiversity Partnership. It's building on a decade of experience from its predecessor, which was Biodiversa. And the partnership started in 2021 and we will run until 2028. It's basically a partnership that was co-designed by the European Commission, so both by DG Research and DG Environment, and co-designed to be a part of the European Biodiversity Strategy 2030, most notably to make the bridge between science, policy and practice. Currently, we are composed of 80 partners from 40 countries, and these are both funding agencies and ministries of research, but we also have on board ministries of environment and environmental protection agencies. So we both have the policy actors, but also the research actors on board, which allows us to better bridge between science and policy, but also to embark on biodiversity monitoring activities. And that's the component that we will focus on today. So it's quite encompassing network going from the north to the south and from the west to the east, and also with a substantial budget, a budget that receives including co-funding from the European Commission, and budget that also includes a big part which comes from the partners as an in-kind support. Next slide, please. So what you see here on the left side, I will not go into too much details, but it's basically the different type of activities that we have within Biodiversa Plus. So it's a broad portfolio of activities. Many of you may know us from the calls for research proposals that we launch on an annual basis. So every year we launch like big calls for research with an amplitude of around 40 million euros to support pan-European research projects. So that's something that we already did in the past and that we continue to do so. But what is actually new for Biodiversa is the biodiversity monitoring activities. And here we mainly aim to promote and support transnational biodiversity monitoring. So the overall objective is to set up harmonized pan-European biodiversity monitoring schemes, but building on existing national and subnational schemes, so not duplicating what is already existing, but rather reinforcing the capacity of the countries for biodiversity monitoring, and also creating opportunities for learning and also for collaboration. So setting up this network is something that we do in collaboration with the European Commission, but also with the GRC, with Europa Bon, with the European Environment Agency, and with other actors, including, for example, also research infrastructures like GBIF. So it's not something that we do alone, we do it in strong collaboration with others. Obviously, we cannot do the harmonization of biodiversity monitoring schemes for like all levels of biodiversity and for all taxa and all priorities. So with the partners, we do have some very specific priorities. And these include, for example, soil biodiversity, invasive alien species, insects, bats, wetlands, habitat quality. So we do have some very specific priorities to focus on. And then we cover a broad range of activities with these biodiversity monitoring work. So it goes all the way from harmonizing protocols, methods, improving data interoperability. But there's also a very strong focus on promoting novel tools and technologies for biodiversity monitoring. That's obviously the component that we will focus on today. And more specifically, we will focus on image-based approaches. But these novel tools, this also includes, for example, molecular methods, acoustics, citizen science, remote sensing, so these type of things. And then it goes all the way down to better use of biodiversity monitoring data, both by researchers themselves, but also by policymakers, and both by private and public policy making. So it's a quite encompassing range of activities. So as I mentioned, ultimately the aim is to set up this network of national biodiversity monitoring schemes. So basically building a network of national and subnational hubs. As a way to also do that, we implement what we call biodiversity monitoring pilots. So these are very concrete test cases, activities that we implement across countries with the aim to establish these long-term biodiversity monitoring schemes, which also includes including some cost estimates. So we also try to make these schemes very resource efficient. So that's a bit in a nutshell what we try to do when it comes to biodiversity monitoring activities. And so, as I mentioned today specifically, we will focus on the novel tools and technologies. And I will hand over to my colleagues from Denmark to introduce you a bit to the specifics of this workshop. So enjoy, and I hope it's a very useful 2 hours for you.
Thank you very much, Hilde. I'm Lars Dinesen, representing the Innovation Foundation of Denmark. I'm going to facilitate the workshop. Good morning to all, and as Hilde has already said, here we have the aim of the webinar. So it's a 2 hours capacity building event where we share information and hopefully enlighten a number of people building bridge between academia, researchers, and people in agencies, implementers, and all other interested. Next slide, please. So this webinar built on a workshop we had in February this year, labeled Towards a Roadmap on Novel Monitoring Technologies and Approaches. And you see the 4 thematic areas we covered at this workshop, and this was a workshop which will feed into the future of the partnership. This is just the first of a number of events dealing with novel technologies. So the others, apart from image-based approaches at that workshop, was molecular methods, acoustic sensors, and remote sensing. But here we will focus on image-based approaches. Next slide, please. And just a brief overview of the registered participants. So as you can see, we have 1/3 or so from research institutions and universities, and we have a great deal from the Biodiversa partners, some involved in the pilots, as Hilde mentioned, and others not, and we have some other interested as well. So as simply we have more than 70 registered participants. I can see we have 58 at the moment, so I think that's really a great number. Next slide, please. So we don't want to waste too much time. So here we have an overview of the program of today. So the first speaker is David Roy from the Center of Ecology and Hydrology in Wallingford. We are very pleased to have you, David, among us here today. And David will set the scene on how to integrate novel technologies in existing biodiversity monitoring programs. And after that, we should have Pierre Bonnet. I hope he's with us to introduce image-based approaches for biodiversity monitoring, and followed by Richard Pucci on how to build image classifiers for species recognition. What are the good practices and things to avoid? Then we'll have a break, and then Tove Thomas Høje from Aarhus University will continue on concrete use of image-based approaches in pilots in the partnership, and we aim to close at noon. So if we don't have Tove is here, I can see. So I don't know if you want to have a few remarks on the pilots. No, I can see. Okay, then we will just go straight to the first presentation by David. Please, David, very much appreciate you are with us here, and please, you have the floor.
Thank you very much. Just share my screen. Yeah, many thanks for this invitation. I've followed the Biodiversa work with great interest and a bit of sadness that the UK is not part of the scheme at the moment, but hopefully there'll be some change in that in the future. Yeah, so I was asked to just give some thoughts on the topic of how novel technologies can be integrated into monitoring programs. And I'm going to really talk about some work that's being done under the Mambo project that Toka leads, and also the Spring project, which is a pilot project to test pollinator monitoring approaches in Europe. So in terms of why the challenge we have of monitoring is that there's increasing demand for information in all sorts of contexts. So we want to look at the long term, we want to look across different areas in terms of spatial planning and understanding how biodiversity changes across different regions. We also want assessments across a range of taxon groups, moving perhaps beyond where we've had good experience of monitoring for some groups, but we know to really understand the environment more fully, we need a more taxonomic spread of knowledge about what's happening to biodiversity, to understand its function, its contribution to people, etc. But that's a challenge for us as biologists and ecologists, because there are many approaches to monitoring biodiversity. Quite rightly, they need to be adapted to the research question or the taxon of interest. But all monitoring approaches are flawed in some way or another. They've all got their biases often in terms of how they detect different species, but also biases in terms of where they can be applied in the landscape, or where they are applied in the landscape in terms of where sites are selected. And there's increasing awareness that this issue of our ability to assess biodiversity is dependent on where and how we sample, and a greater call to be more explicit in some of these biases and how we deal with them, but also communicating them. So there's a paper here published in Tree recently from some of my colleagues, which talks about some of these issues. And I mentioned that we ideally want a broad assessment of biodiversity, but we know that not all taxa are given equal attention in terms of research effort, monitoring schemes, conservation effort. And this is illustrated by this figure here, which is taken from a paper a few years old now, but I think the story, I'm sure, is still the same, in that if you look at data through GBIF in terms of the occurrences being shared through the global platform, compared to the number of species in that group, we have a predominance of data for birds and mammals, for example, shown in the green bars. And at the other end of the gradient, we have insect taxa, other invertebrates, some lower plants, for example, where we have much fewer data, but a high diversity of species. As we know, there's around 1 million insect species described, and it's been estimated there's 10 quintillion individual insects at any time, at any place, at any time on the planet. So they're both incredibly important in terms of their diversity, but also their ecological function. So if we really want to understand the environment and biodiversity, we really need to be paying more attention to insects. So it's really great that there's this increase in options for monitoring insects, particularly through new technology, summarized nicely by this paper by Van Klink et al., which focuses on 4 main technology areas: computer vision, acoustics, radar, and molecular methods. So I'm going to say a bit about molecular methods, but the focus today is mostly on computer vision. So I'll focus on that more details. And I think these new technologies allow us to do new things in new ways and generate new data streams, but they also allow us to do things more efficiently. So I'm just going to give an example from the pollinator monitoring world. This is related to the Spring project that I mentioned that's currently working across Europe, funded by the European Commission. But we've also got a longer experience of this monitoring through the UK pollinator monitoring scheme. So common to both challenges really about how you monitor pollinators across Europe is that you generate a huge amount of material. So the typical, one of the main methods is to use a trapping method, water traps, or pan traps as they're also called, which are put in the field for a day. So they collect insects that fall into the liquid and die. So they're then collected into tubes that are then posted to a lab. And then there's a sorting process that separates out the pollinating insects from the rest, because the main focus is on the pollinators, the bees and hoverflies. But we also have a lot of what's called bycatch, a lot of other Diptera, Coleoptera, etc. So a huge amount of material that needs to be sorted, but then it also needs to be identified. So typically that's done by taxonomists under a microscope, hugely expert people. So there's an issue of time to do that, but also capacity in terms of the number of individuals with the expertise to do this. So molecular methods are a potential solution, and there's some promising results. But molecular methods will give you certain types of data that's different to a sorting and taxonomic approach, where you'd get better information on the abundance of individuals. But also imaging has a potential role in this to help with some of the sorting and identification. And thanks to Toka for this slide, which shows some nice approaches using a camera setup, the bioscan system, where individuals are put into a mechanism and float through ethanol with a camera focused on the insects as they fall through the liquid. So giving 3-dimensional views of the insects, which can then be supported by machine learning for identification, and supported by robotic techniques to sort the sample, could potentially be add great efficiency to this process of manually sorting and identification. So that's a sort of an story of efficiency through molecular and image-based approaches. I also want to talk about the second area of how to integrate new technologies. Is, as I mentioned, they're likely to generate different data streams and have strengths and weaknesses compared to traditional approaches. So the challenge we have is how we bring together these two different sources of information to answer the same question. So there's been some statistical development in this area. Again, a tree paper led by Nick Isaac, where it recognizes that the thing of interest, or the state of the environment, is what we're interested in. And we have multiple measures of that state. So this can come from different sampling schemes. And we can use statistical models to reflect the differences in the sampling methods and their biases, and correct for their how they detect species, but then bring them together in terms of looking at how the state, the biodiversity state, is changing, for example. And you can think of this in terms of all different sorts of scenarios. I think this is going to be a common issue that we have when we try to integrate new technologies with traditional approaches, that we're not, I don't think, we think that new technologies will replace other techniques. I see them as very much being complementary. So in the example of the pollinators, again, the 2 primary methods are the pan traps that I illustrated before, but we also rely on transect walks, where we can involve citizen science, for example, but also experts who could identify species in the field. And the two different methods have different advantages and strengths and weaknesses. The pan traps is typically used for presence absence data and species richness, because the abundance of insects can be highly influenced by the landscape in which they are put in terms of the flower density. But transects tend to be more suitable for abundance measures, counting number of individuals. So they're measuring different aspects of biodiversity and using technology, as I said, to help sort the pan-trap data. And image analysis can also support the transect recording. These can be brought together into a single model which borrows strength across the two different approaches. And a second example of this we can think of, and that we're also testing within various projects, including the Biodiversa Plus pilot, is to consider how automated camera systems, the Amy trap developed by the Aarhus team and now being used in many places across the world. It's providing an automated assessment of night flying insects. We've also got more traditional bucket traps or interception traps where citizen scientists or professionals may put the traps in the field, collect the individuals, and then identify them, again using camera systems or image analysis to help with the identification. So what we're doing in the Mambo project is we recognize that there's going to be a lot of innovation in this space, a lot of new sampling methods developed. And as I said, we see these as being complementary to traditional approaches. So at the moment, we don't have many examples of where we've got the traditional approaches and technology together. So we're going to use a simulation approach where we will simulate data that would be collected through different sampling protocols. Here is illustrating an example using technology and transects to then feed into these integrated models to then compare the simulated data to how the models produce. And what's also interesting here, we can also think of this in an adaptive way, that we also need to consider where in the landscape we tend to use technology versus traditional approaches so that we can direct effort to where it's added most value. So ideas around adaptive sampling and to redirect sampling to places where we need information most effectively. So we can think where can we base both best placed technology solutions, and where do we need then citizen science approaches to complement that monitoring so that together we have an integrated assessment of biodiversity using both methods most efficiently. So in summary, I think there's increasing call on monitoring data, particularly providing evidence for policy and how we meet targets. There's a big increase going on in terms of monitoring approaches and data coming through monitoring systems. And I think a new technology significantly adds to our toolkit to both monitor the environment, but also how we analyze and process samples. And I would argue that this integration is one of the key tools that we need to develop to make best use to maximize the value of these multiple data streams. Thank you.
Thanks very much, Roy, for a great presentation and for highlighting also the need of focus on some of the lesser known biological groups such as insects, and telling us about the Mamba project. And I hope you and UK will join us soon again. I can see there's some movement going on, but let's see. So this was really a nice setting the scene, and we have not programmed any questions, so I will continue to hear if we have Pierre Bonnet among us. I can see you. Pierre is here. That's great. So Pierre will introduce us to image-based approaches for biodiversity monitoring, and you're coming from Montpellier in France, and is an expert in plant science and the application of computer technology among others. And I guess you will share your presentation from your computer, I guess.
Yes, let me know, Cécile, how you would like to proceed. I can share my screen.
Maybe if you can share your screen, that's okay. Yeah.
For sure. Can you confirm me, Cécile or
we
Lars?
can
Yes,
see the
as you
full
can see
screen.
it.
Perfect.
Thank you very much. So thanks a lot for this opportunity and this invitation. So as it was mentioned, I'm based in Montpellier in a research lab focused on tropical botany. I work since a long time with a research team in computer science in Montpellier. And so this presentation is based on interaction between ecologists from my labs and colleagues working with us, and a computer scientist and engineer who work with us in the development of computer vision methods and application of deep learning for ecological study, mainly focused on plants, but not only. So the massive production and availability of multimedia data, which are acquired now at a very huge scale through the decreased cost of sensors that allows to produce multimedia records in our environment, increase the volume of visual data that we can now engage in our research activities. Similar, we saw an increasing capacity of computational resources at even regional, national, and European scale. And all these new resources allow to store, manipulate in a new way this huge volume of data, and the growing social demand for automated tools to analyze the visual data as a push major advance in image analysis over the last decades. In order to measure the benefit of this improvement, since more than 10 years in our research group, we conduct an international challenge which focus on the evaluation of computer vision and machine learning methods for plant species identification. So this challenge entitled Plant Clef is organized since 2011, and it allows us each year to share visual data sets with a computer science team interested to compare their approach to the scientific community. And so what you can see here is the best performance obtained by the winner of the challenge each year since 2011. And what we can clearly see is progress of these techniques in terms of accuracy and performance for this task that we organize. And the fact that even if we increase the complexity of the task, so on the table on the top of the screen, you can see the increasing number of species and images engaged in the challenge, up to 10,000 species illustrated by more than 1 million images in 2017, we can see a clear increasing of the performance of the methods that have been tested. It was clear in 2015 that deep learning methods, which is a class of the method in computer vision, clearly overcomes the other approach. And since more than now 7 years, most of research activities focus on the development and the improvement of computer vision, embed in some way the use of deep learning models. So computer vision is at the intersection between machine learning and signal processing, and it deals with the extraction of information from digital images. So the automated or semi-automated extraction of such information. There is a wide variety of methods and tools available, and the careful selection of the most appropriate one according to the data available and to the research question that we want to invest is a critical step at the beginning of each of our new experiments. Lurig illustrated here diversity of methods with a gradient from small to very large data sets, and from standardized to less standardized and more noisy visual data sets that can be used. And what we can see is that 10 years ago, signal processing techniques were applied on a quite well standardized images for a data set of quite small size. We saw the arrival of machine learning techniques due to the increasing volume of the visual data sets that could be of interest for research in ecology. And since now less than 10 years, the availability of graphic processing units and very large visual data sets allow us to take advantage of deep learning techniques that allows to be much more efficient in a context where we have to deal with large but noisy visual data sets. And so in order to illustrate that, I come back again to this international challenge that we organized. And in 2017, we did an experiment where we asked to the participants to evaluate their methodology on a common data set, but the data set was divided in several subgroups. So we have in green visual data sets that was dedicated, that come from research institutes. So it was a quite small data set of several thousands of images illustrating 10,000 plant species. And this data set was available with another one illustrated here with a method trained on that data set, which was a data set coming from a crawl of the web using a Google search engine and Bing. And what we can see is the result of the methods trained on a trusted training set, on a noisy training set in red, and on a merge of both a noisy and trusted training set. And what we can clearly see here is the best performance of the methods that took advantage of both data set, noisy and trusted one. And so this is mainly due to the fact that all the techniques evaluated here were based on deep learning methods which deal well with such contexts in terms of data. Another illustration that we provide here on the same challenge allow to see between the performance measure for yellow and orange, the gap that we have between methods that were trained in 2014 and method trained in 2017. And so this method clearly show a much better performances few years after the first challenges. And what we can see here is a comparison between orange and green of the capacity between human in green and orange in for automated approach to well deal with a species identification based on image analysis. And what we were able to see here is that even a human expert cannot reach 100% of correct species identification based on image analysis due to the fact that visual content are not always present in and informative enough to recognize correctly a species in an images. And automated approaches are very close to human experts, even for some of the most difficult plant species that we have in our region. So due to the performance of deep learning methods in many different domains, their use have been evaluated in different ecological contexts and focusing on different type of data. So Borowiec illustrated here different type of data that we used to collect as ecologists. So the first one is, for example, digitized herbiome specimen, but we could also have the same type of visual data related to field plant or animal observation produced by citizen scientists or autonomous sensor. Songs are recorded through the use of record that allows to transfer this song in a sonogram, and which allows the application of deep learning techniques to eventually characterize bioacoustic environments. The third illustration here, which come close to some of the subjects previously introduced by David Roy, illustrates the potential of the use of this method for the analysis of insect motion with a fixed camera. And so, as you can see here, the multimedia data that are generated are strongly correlated to the sensor and the protocol of collect, which is engaged. And it is often important to well think the use of these methods in a workflow that come from the collect of the data up to the research question that we want to target in the end. And so here, Christine illustrated some scale and type of application that took benefit of the use of deep learning techniques applied mainly to visual data. So such application include a recognition of individual animals. These have been mainly experiments for the monitoring and track of individual mammals, the analysis of population and group of plant or animals, some analysis that have been conducted at the landscape or ecosystem, but recently we could see the potential of such method even for global assessment. So in order to successfully apply computer vision technique, and in particular deep learning one, a number of practical considerations are important. So it is important to think from the beginning of the quality and volume of data that we can reach according to the research questions that we want to consider. It is also important to think that there is a need in investment of manual annotation in order to generate a ground truth for a subset of the data that we want to use, which will be dedicated to measure or detect error or potential bias in the methods that we will experiment. And then the choice of the methods to analyze such data is quite important, and it's important to take into account their computational and technological costs. So this means that some techniques, even if they are very efficient, they are quite data intensive or computational intensive. And so it is not always reachable, we could say, due to the expertise that is required to mobilize them. So I will finish this presentation with some concrete illustration on the platform that my research team manage. So the Citizen Science PlantNet platform, which is dedicated to make plant recognition based on image analysis. So the network of end users involve a large volume of citizen scientists, even ecologists. And so the full framework that we designed was dedicated to increase the capacity of citizens to generate high quality images based on the use of a deep learning model, which is continuously trained according to the new volume of training and validated data generated by this network of users. So we deal with different type of deep learning models. And so here below you have the top one accuracy for 2 deep learning models that we have experimented. At present time, the platform rely on visual transformer, mainly due to their better accuracy for plant species identification. But it was a quite big challenge for us to move from Inception V3 model to Bait 1, because the computational resources consumed for such large model are quite important. And so it took more than 1 year for us to do this migration according to the computational costs that we have to invest in order to allow this change, even if you can see the difference in terms of accuracy is quite small. It is important for us to take into account the uncertainty in the predictions that are generated by our approaches, and there is 2 types of uncertainty. So one, which is the ambiguity, which is irreducible due to the fact that the visual content recorded in an image is not always enough to correctly recognize a species. And the second one, illustrated on the right here, which illustrates the fact that the most well recognized species are the one for which the highest volume of training data are available. So we know that some species are much less well detected and recognized compared to others. This mainly related to the difference in the volume of training data that is available. So this is why we engage the users in the production of the new training data and in the revision of the quality of the data. So we have implemented some mechanism related to cooperative learning in which end users of this infrastructure, of this system, clearly participate to its improvement for their needs. Among all the data that we aggregate, so we have received more than 500 million of identification requests. Only half of them are geolocalized. So this means that only half of them are of interest for research question in ecology. But we want to invest in the use of the generation of visual data in order to allow us to increase plant species identification based on this new training data. So a fraction of this data are shared on GBIF platform. So more than 13 million of plant occurrences are shared. And we have a pipeline that allows us to filter all the data, all the raw data aggregated at the level of the platform based on visual analysis in order to extract the most interesting one for the GBIF community. Due to the complexity of the infrastructure, our choices are strongly related to the efficiency of our IT capacity. So this means that we have to take into account and to deal, for example, with the image size, the time to process the different type of data and different algorithms in order to ensure that we have a real-time system that is available and accessible to all our partners. Recently, we designed a new functionality at the level of the platform, which allows to embed the deep learning model on the smartphone itself in order to allow people to make species identification in a remote condition without connection. And the choice of the deep learning model to do that was a key criteria to allow to have efficient offline model. And I will finish with this illustration, which give a broader picture about some of the research questions that we invest based on this data and this method. So here is a proportion of endangered orchid species at the world scale and at the resolution of 1 kilometer. And most of the training data used to generate this map at the world scale are based on GBIF data, and we use what we call a deep SDM, so the use of deep learning techniques that have been trained on satellite images in order to predict proportion and distribution of species and proportion of the endangered one for the orchid family. Thank you very much for your attention.
Thank you, Pierre, very much for a very interesting presentation and for highlighting the rapid development within this field and bringing us from the individual level to ecosystem and global level, and also even with artificial intelligence to highlight that there's still some insecurity in identification, although the computers have beaten us, it clearly seems. We have a few minutes for questions, so please, if there are any questions for Pierre, you're welcome. And I think Cécile, you will check the chat, and otherwise you can raise your hands if you have a burning question. I see Tove, please.
Hello, Pierre. Thank you very much for the excellent presentation. Clearly, this is a fast-moving field, and the data generation happens very fast. You show this from all the work you've done and these tools to engage the general public in collection of data. is really exciting. I wondered if you could reflect a little bit on how you see the future of this field, receive data from automatic sensors compared to what extent you think like smartphone based applications will continue to be the primary data source for the kind of tools that you're describing.
Yes, so I think that according to the discussion that we have with the partners involved in the generation of data through these two complementary ways, what we can see is that when you use an automated sensors, which is not in the hand of somebody, usually it's usually dedicated one that has its own capacity, artifact, and that is able to generate a quite huge volume of data, but it's quite difficult to manually and collaboratively analyze such type of data. So this is sometimes feasible for particular experiments, but most of time, due to the very huge volume of visual data generated through only one drone flight, for example, or one camera track embedded on any autonomous engine, due to the huge volume which is generated here, human is difficultly involved in the loop of the analysis of such data. So we have to develop new pipeline to automate their analysis and to only identify the key type of analysis that have to be manually done in order to improve systems which are based on such data. So I think that probably the mechanism related to active learning, cooperative learning that involve a human evaluation and validation on this data generated through autonomous sensor will be a key criteria to allow us to take benefits of such new type of multimedia data that can be generated at a huge scale, but that will be unuseful if we are not able to correctly track their interest and improve their interest for our research question.
Thank you very much.
Thanks, Tove, for the question and for lengthy answer. Before we continue, I can see there is a question from David. Please, David.
Yeah, thanks, Pierre. I had a question about the limits of image-based analysis for plant species, because we know that some plant species are very difficult to identify and need to look at fine-scale features. Have you had an assessment of how far you think you can go with the image analysis, whether they'll actually prove to be quite effective where traditional approaches are less suitable?
Yeah. So clearly, visual analysis cannot alone solve the problem of identification. This is for sure. So we have to combine it with several other factor, criteria, type of data. I did not mention that, but I think that geolocation is a key feature. And there is also several other type of information that can be recorded for a user on a user point of view, but also there is other type of automated data that can be processed. So even if LiDAR start to be embedded in smartphone, and even if there is a huge volume of work that have to be conducted, we will be able to collect much more information related to the size, texture, shape, and not only collected through visual approach, but from other sensors that will be embedded in smartphone and which will allow to increase the capacity of automated approach, but taking into account a broader group of type of data, I think. Yeah.
Yeah, great. Thank you.
Thank you, David. Thank you, Lars.
Thank you so much. Thank you. And then we'll continue to...
maybe Lars, there was just one question in the chat from Dietmar Moser. For which taxa work this method best? So I don't know if maybe Pierre would like to answer this one.
Yeah, I think for plant groups, what we could see is that for plant species that have a quite broad reproductive or vegetative structures, so such as big trees, it's much more efficient than for grass and plant with a very thin and light and less compact visual organ.
Thanks, Cecile, and thanks again, Pierre. And then we will continue to welcome the next speaker, Rita Pucci from Naturalism in Leiden in the Netherlands. And Rita will speak about how to build image classifiers for species recognition. What are the good practices and things to avoid? And Rita is a postdoc and researcher in machine learning and image-based identification, among others. Most welcome, Rita. The floor is yours.
Thank you for inviting me. So let's share the presentation. Oh, no, I cannot do that.
Or if you want, I can also share your slides. It's up to you.
No. I have double screen. That is the problem. Okay, let's do it this way. Okay. Can you see my slides?
Yes, it's full screen.
That's good. Perfect. Okay. Hello, I'm Rita Pucci. I'm a postdoc in deep learning for computer vision in Naturalis. I work in Mambo. And today I'm introducing to you some concepts, more theoretical, I have to say, because I'm new in this field. So we start with some vocabulary and then some history of deep learning for computer vision, and then some good practice and some problems when we talk about classification of species. So first of all, vocabulary. So I'm always listening people saying to me, you do artificial intelligence, we can apply artificial intelligence, but artificial intelligence is a lot of things. So artificial intelligence are all the algorithms that can make a computer do something that normally is done by a human. So whatever, from robots to surveillance, everything is artificial intelligence. But in this broad amount of algorithms, there are some algorithms that are using just imaging and videos. In that case, we talk about computer vision. And there are some algorithms that are instead focused on the learning procedure. So emulating the learning of humans in case of a new task or concept. And there is an intersection between these two fields that is machine learning applied for computer vision. But obviously, nowadays, we don't talk anymore about machine learning for computer vision, more about a subset of machine learning algorithm, that is the deep learning. That are a type of machine learning algorithm, but where the structure of the algorithm is resembling the structure in some way of the human brain. That means that it is deeper, is huger, and is more powerful. And we are interested in this intersection. So we talk about the deep learning algorithm for computer vision. So this intersection, I want to start with a rapid trip in the fields of the green areas, so the computer vision and the deep learning algorithm. So we start back in 1958, when there is Humbert investigating vision reaction in cats and discovering that there is a hierarchical structure of cells to recognize the stimulus for light, movement, and endpoints. So different cells, simple cells, complex cells, and hypercomplex cells that are recognizing completely different things. And these things are building the receptive fields for vision. So that is the first work that is interested in understanding how vision is working. Then in 1963, Roberts studies, okay, but how can I see an object? So the first thing is, can I see edges of a solid? So edges are really important to identify where the object is in an image before everything else, and which is the shape of this object. And that is an important thing, because if we see edges, as human, we recognize edges naturally. But for a machine, to recognize edges is an important task and not so simple. But okay, but how to identify the same object if I have the same object in different orientation in different images? Then it's coming a new concept in vision, that is features. So then in 1999, answer this question with, we need features. So properties of the object that are invariant completely or partially to this modification. So to scaling, translation, rotation, or to illumination and 3D projection. That is a fundamental point in the history of vision, because features are always used for image recognition. And they are, from 1999, we were able to do face detection, because we were able to recognize shapes, and we were able to recognize the features of something like a face. Then in vision, in 2007, the researchers were interested in collecting as much as possible samples of categories, so we could understand the features of different objects. So in 2007, we have ImageNet, the biggest data set that was proposed, 1,000 categories, more than 1 million images, and is the starting point for everything that is now deep learning for computer vision. Then we move on the other side. We have machine learning. That is another thing from vision. Ed was doing his stuff. So machine learning started with, okay, we have, at the beginning, we have a similar start with structure, the hierarchical structure of feature extractors connected. That was extracting what we needed to gradually extract to understand what is inside the image. So from a little part of edges, then compose them, then the entire object. So features and hierarchical structure are coming also in machine learning. But then what? Then we need to learn, because it's machine learning, so we need to learn something. So we need to learn to extract these features. That we need these features to recognize the object in images, and so we need a learning procedure. That this learning procedure was introduced in 1987 by Rumlot, and is based on feedback propagation, like humans. So my algorithm is predicting something based on the connection of the structures of features. And I'm saying to him if it's correct or not. And if it's not correct, he has to change the values of the connection in order to predict the correct answer next time. So every time that my model is predicting something, I'm giving the feedback. And that is the basic of the backpropagation algorithm. That is not the variation of that is still used nowadays for machine learning algorithm. Then in 1998, the most famous structure for image classification is proposed, that is convolutional network. That is hierarchical, is extracting features, and is trained with backpropagation. So we see as machine learning for computer vision, deep learning for computer vision, because now convolutional neural network, they introduced the concept also for deep learning, is not a black box. It's a long process started in 1960 and arrived at the end in what is now really famous as deep learning. So that was an introduction just to understand what we are talking about. Then now we move in how to create a classifier. There are three keys that we have to take in consideration. The first thing is the data set. So the data set, as we saw, has a huge role. And we need a data set that can be split in 3 data sets. The first is the training set, so the biggest one. There is an order of thousands or billions of samples. In case of ecology, I have to say thousands. Then we have a validation set that is used to evaluate the model while it's training, so during the feedback procedure. And then we have a test set. So the test set is completely external. So the best practice is that the test set is coming from another group that is doing the same thing, and so we can test our model with other data sets, but it can be also our test, but it has to be completely unknown for our model. So that is the first thing. We need data. Then we need a task, obviously. Well, in this, there are multiple tasks. But in this case, we focus on the classification task. And for the classification task, there are 3 types of tasks. So the multi-class classification. So I have this image. I'm saying, among all the category of animals, that's a cat. Then I have a binary class that is say, okay, is answering the question, there is a cat inside. And I said, yes, there is a cat inside. And then I have the instance classification. So I have an image of 2 cats, and I want to identify both independently. There are cat number 1 and cat number 2. So we focus, for species classification, we focus on the multi-class classification. And then we have the giant gap between human and machine. For human is a beautiful picture of a cat. That's my cat also. But for machine is not that. For the machine is a giant grid of number. So everything that could be obvious for us is not so obvious when you see it as just a giant grid of numbers. And also, usually the images are RGB. So I have a giant grid for numbers, 1 for red, 1 for green, 1 for blue. So if I have this image that is 800 by 600, I have a giant grid of number that is 800 by 600, 1 for red, 1 for green, 1 for blue. And depends of if I'm talking about RGB or others. In case of RGB, I have this number cannot be whatever, but has to be between 0 and 255. That is the definition for RGB. So when we talk about images, we have to take in consideration that that is what the machine is seeing about an image. And then we have the challenges, because it's easy to classify, for example, a cat. Well, not always, because a cat can be seen from different viewpoints. Still, the cat is my cat, is the same individual, but it's completely different from an image to an image. You have to take in consideration that are numbers, and here we have completely different colors for the same cat. But we have also different breeds. So still cat, but completely different appearance. And then we have also the deformation. So a cat can appear completely different from an image to another image. It's still the same cat. You have to trust me because that's my cat, and I know that is the same cat. But can you imagine the difficulties of seeing this cat in these 3 images that seems to be completely different things? Then we have the illumination. Sometimes the cat can be completely black or completely white, but it's still the same gray cat. We can have occlusion and camouflage. So that is the first challenge that you have to take in consideration. And yes, it's important to check our dataset for these challenges. We have to be sure that our model is trained on all the possibility of the appearance of our class, well, all, as much as possible. And we have to take in consideration that it's not always so simple the task that we are asking, even if it's classification. And then we have another type of classification that is still multi-class, but it's deeper. So we had the fine-grained classification. So on top of this problem, when we talk about fine-grained classification, we are talking about subjects that are really localized, like the breathing cat. So in my case, I'm taking consideration species. So the taxonomical concept based on the hierarchical structure of the taxonomic name. And when we talk about species, we are talking about images from the same order of animal. In this case, I'm taking Coleoptera. The same family, in this case, Coccinellidae. And then I want to discern between the species that are really similar. But then I have another problem with fine-grained classification, that is the long tail distribution. So I have some species that are really common. They are the head of the class, so I have lots of samples for them. And I have some species that are really rare, the tail of classes. So for them, I don't have so many species. So for this problem, so if for the challenges before, their solution is to have as much as possible samples, in this case, we don't have samples and we have to generate new samples for the rare species. So we can apply some data augmentation techniques that are normal for machine learning and deep learning. So data augmentation techniques are techniques applied on directly on images, and the goal is to generate new images. They are similar to the original image, but they are changing in something, and for the model is enough. So we can have a shift of the image, we have a rotation of the image, saturation, color augmentation, but there are lots and lots of techniques. Everything that could be applied on the image can generate a new image. But in this case, we have to be aware that not all the augmentation techniques can be applied, because if we are doing fine-grained classification, we cannot change the colors, because colors are part of the definition of the species, or we cannot change completely the rotation. Can we not do the upside down image? Because sometimes the image is with no sense when it's upside down. So there are solution techniques, but we have to take in consideration which techniques we want to apply. And then there is the big question, which model? So the model question is a huge question, because models nowadays, they are super powerful, and is really a thing of 0.3, 0.1%, that is the improvement. And as said, the important is to take in consideration the inference time of the model, or also the computational requirement of the model. But there are two families that we are taking always in consideration. One is the convolutional model, that is extracting local features. So the convolutional model is using a kernel, as we see in this GIF, that is extracting patch by patch features of the image to generate a sort of negative of the image, and that is the negative of all the features of the image. The more that we are going deep in the structure, the more that we are refining the feature that we want to keep, and that we learn that are useful for the classification. On the other side, we have the transformers. So transformers are really new models for computer vision, and they are starting from a completely different concept. Is not, I don't want to extract features as a negative, but I want to analyze the relationship between patch and the importance of the patch for my classification. They are coming from natural language process, so you have to think that a transformer is built to be for a phrase, so a phrase with words. So when we want to give an image to a transformer, the first thing that we have to do is to transform the image into a phrase. So an image is split in patches, and each patch, that is a little square of the image, as we see, each patch is a word for our transformer. And with some mathematical process, we change from the square definition to a vectoral definition, so a linear list of numbers. And in the transformer encoder, we are generating the attention among all the patches. But the important things to take is that when we use convolutional, we are understanding local features. So in the bird, we are analyzing the relationship between the eye and the beak, but not with the eye and the tail. Really difficult in that case because they are far apart. But when we are taking consideration transformers, is everything with everything. So it's a global extraction of features. So we are trying to globally in the image what is important for the classification. So to answer the question, which model? It depends. Sometimes can be good the convolution, sometimes can be good the transformer. So for example, in my case, I have to do fine-grained classification of insects, and I have to decide which model. So the first thing that I did was a comparison among models to understand which are the skills of each model. So I took the Coleoptera dataset that I have with 3,087 species and 178,000 samples. And I tried to understand the behavior of 3 different models when we have different amount of samples per species. So I took the FisherNet V2 as convolutional model, the token-to-token vision transformer as transformer model, and I wanted also to compare with the new class of models that are the hybrid model. In this case, I took the Vision Transformer Advanced Efficiency that is taking consideration both convolutional and transformers in the same structure. And as we see, if we have a lot of samples per species, the models are almost with the same performance. It's really 0.3 or something like that. But when we go to the species with really low amount of samples, even if obviously the accuracy is really low because it's like 15%, the maximum, we can see that the hybrid one is trying really hard to have a better accuracy. So we can imagine that a hybrid model can get more details about image, about each image, and so is able to generalize better in the species. That could be an interesting point to start with new models. So I did this comparison in the context of MAMBO. As we saw, MAMBO is a huge European funded project, Horizon Europe. My project started in 2022 and is based on model approaches to the monitoring of biodiversity. What I'm doing in this project is taking all the images collected by citizen science through the app Obsidentify. These images are stored in observation.org datasets, and I'm taking these images and I'm now analyzing new models, modern models, different type of models, to understand which model is performing better or which is the behavior of other structures in the context of fine-grained classification. The fine-grained classification in this project is applied for biodiversity protection, conservation status monitoring, threatened species monitoring, and ecological requirements understanding. If you are new to classifiers for images, there is an interesting tutorial that is based on Python and on the library PyTorch for machine learning. You don't have to install absolutely anything. It's completely online. It's provided by Google. And you can try to use convolutional neural network and to see how it's performing, how it's not performing. All the material in these slides are available on this bibliography. If you are interested, you can read these papers and discover more. Thank you for your attention.
Thank you so much, Rita, for a very interesting presentation, and we really a lot to digest here. You brought us on a historical tour and discussed the terminology and continue with all these issues on data classification and which model to use and so on. Thank you very much. We have a break soon, but if there are any quick burning questions, we can take them on board before we take the coffee break. I don't see any at the moment. I don't know if there's any in the chat.
No
In
question.
case, I can also receive emails with questions when you digest everything.
Sounds good. If there are no questions, then we will take the coffee break, and we will join again in about 15 minutes, 11:30, and be ready for the presentation by Toge on some concrete examples. So please come back 11:30. So I think we can start up again. I hope you are back from your coffee break, or grabbed a good cup of coffee or tea. And the next presenter is Toge Thomas Høyer from the University of Aarhus, and leader of the Mambo project, which we have heard a bit about today this morning, and also lead of the monitoring pilots in the partnership. And Toke will give some concrete examples of use of image-based approaches in the partnership and in the monitoring pilots, which is leading. So Toke, the floor is yours, please.
Thank you, Lars, and thanks to all the speakers so far for setting up this scene so nicely for for the concrete activities that we do in the pilot scheme under Biodiversa Plus. So yeah, as Lars said, I represent the Ministry of the Environment Denmark as coordinator of this pilot scheme, which was launched this year and is now continuing into the coming years as next slide, please. As a way to test the deployment of key elements of biodiversity monitoring, some of which involve more modern technology and others which are trying to harmonize existing approaches to biodiversity monitoring. In Biodiversa Plus, through the partnership, the pilots are also a way to come together on concrete topics to share experience and to build capacity when it comes to biodiversity monitoring. They are not really seen as a research project, but more as a way to test more mature approaches, but not yet generally implemented approaches to biodiversity monitoring. And they are either focused on species monitoring, habitats monitoring, or more broad or transversal in terms of the management or governance of existing programs. Next slide, please. So at the moment, in 2023, Biodiversa Plus is running 3 sub-pilots or elements of this pilot program. One on monitoring invasive alien species, plants and insects, one on soil biodiversity in near natural forests, and one of these more transversal activities exploring the governance structure of existing national monitoring programs and their data interoperability and data standards. Just recently, the General Assembly of Biodiversa Plus has agreed both to extend the first 2 sub-pilots on invasive alien species and soil biodiversity into 2024, and to establish 3 new programs, sub-pilots, one marine on European rocky fish monitoring, one focused on images and sound recognition for monitoring of nocturnal insects, birds, and bats, and one on habitat monitoring, primarily focused on remote sensing and the ability of remote sensing tools to monitor specific aspects of the extent and condition of grasslands and wetland habitats. Next slide, please. So as you can probably tell, there are a number of these sub-pilots that take image-based approaches to biodiversity monitoring. 2 of them relate to specifically to image-based approaches, the invasive alien species pilot, which is ongoing and for which I'll spend most of the time here talking about. But as of next year, we'll also be using image-based approaches in the ABMS sub-pilot on automated biodiversity monitoring stations. So I'm showing you a map of the countries or locations where partners are currently involved with monitoring species with image-based approaches. And this is the map of the partners involved with the invasive alien species sub-pilot. There are 10 partners involved, and they take part in both a plant module and an insect module. And as I said, that this was originally a 1-year project, but it's now extended to next year, and additional partners are welcome to join the activities for 2024. Next slide, please. Yeah, more specifically, these two modules are operating two types of camera equipment to record images that are then fed into subsequent analysis. For plants, it's a custom-built high-speed camera that is able to record images of vegetation along roadside verges at driving speeds up to 110 kilometers an hour. So these are cameras that are very light sensitive and are able to record sharp images even at this speed. And this obviously allows for rapid generation of images as the car moves through landscapes. The other module is an insect camera trap that is specifically targeted at nocturnally active insects that are drawn to light. So it is fitted with both a UV light to attract insects and visible light system to illuminate the insects for recording. So I'll go a little bit more into detail with the types of data that these systems are generating and how we are working with this data to extract actionable knowledge from them or meaningful ecological data. Okay, next slide, please. So for the plant module first, here's one of the images recorded with the system. Yeah, I believe probably this was recorded at 90 kilometers an hour. You can quite clearly see the lupins in the front of this image, and generally the image quality is quite high. This obviously means that we have quite a lot of data generated. These cameras record about 5 frames per second, and we're building this system to allow a co-pilot to tag images when key species are being detected along the roadside verge. So we have a way to introduce a preliminary expert-based filter on the image recording, but the camera systems are recording continuously, so we can then later predict the occurrence of key species across the entire road stretch that the cameras were operating. And next slide, please. In the image analysis side of this work, we're working closely with Pierre Bonnet and his team, or the PlantNet organization, to build upon the work that he spoke about earlier on the PlantNet image classifier. This is a classifier that only returns a probability for species within an image. So originally the classifier is built to only take full images and not do any localization of where the plants are within the image. But together with the PlantNet team, we've now come up with a solution that allow us to localize these plants more specifically within the images. And this is done by kind of a multi-level inference, where we cut out various subsets of the image with different image sizes, and feeds each of these subset of the image to the image classifier in PlantNet, and extract a probability of the occurrence of each of the target species that the partners have focused on in their image recording, to allow us a synthesis across all these various spatial scales. So in total, each of the images that are recorded with this system will be cut out in many different smaller subsets, and each subset is then submitted to the Planet classifier. In total, 328 classifications are thus made on each image, and we can synthesize the probability on the scale of the individual image as to where that particular species, in this case, Osa rugosa, would be found in the image. So not only does it give a probability for a species to occur in an image, but also to where in the image it would be found. And this helps us in the expert review of these classifications to have this localization. In total, this takes around 20 seconds when we do the automated submission of individual images. So it's still tractable to analyze large numbers of these images, even with these many calls to the classifier. Yes, next slide, please. That's the stage where we are at right now. We've been collecting images across these 10 countries this summer, and currently this image processing pipeline is running day and night, basically, with the images collected by the partners. And once that process has come to an end, what we envisage is that we get information to be able to create maps such as these that are not created in the Biodiversa Plus pilot, but by a national project in Switzerland that already had worked along similar lines a few years back, and these visualizations will then allow future managers to be able to quickly learn how the probability of key invasive plant species change across the road network and where to direct their efforts. So here are multiple images of the same road stretch recorded at multiple different time intervals at both driving directions on a particular road stretch. So imagine we will ultimately be able to present such large-scale maps for the areas where these cameras are operating. Yeah, next slide, please. For the insect module, we are obviously in a position where the imaging conditions are much more controlled. The nocturnally active insects are attracted to light, and the camera is already in place in front of this screen. So we have a very simple background against which we can image the moths and other insects that land on this screen. And here is just one example of these images recorded with the system that illustrate, well, first of all, how numerous the insects that land on the screen are, that in any particular moment in time, there can be quite a number of individuals sitting in front of the screen. It also illustrates that some of these will not be sitting, but will be in fact flying. In the lower left corner, you can see that some moths will be quite active around the trap, and therefore we will also detect animals that are more difficult to classify because they are in free flight. And it also illustrates that it's not only moths that land on the screen. There will be a lot of small, typically Diptera, but also other taxa that arrive to the screen. And while this is ecologically interesting and relevant for this invasive alien species subpilot, we focus on moths and those taxa that were identified by their partners to be of key interest in their region. What you can also see in this image is the result of both a localization algorithm and a classification algorithm. So localization means that the machine learning algorithm has been able to localize each of these moth individuals and put a bounding box as to where they are. You can see that in some cases there are actually some errors in that. In the bottom center of the bottom, you will see one bounding box that actually encompasses 2 individuals, and that happens a few cases with this earlier version of the localization algorithm. This has now been improved and is occurring less often. And you can also see that there are species names and percentages associated with each of these bounding boxes, and that is a classification algorithm or the result of a classification algorithm that gives us some impression as to both the likely species name and the certainty that this species name is correct given the classifier. We'll come back to that in a little while. So next slide, please. So broadly, the approach that's taken in operating or working with these insect camera trap images is to split up the task of localization, finding the insects in the broader image, and classifying them first into broad groups of moths and other insects, and then doing the fine-grained classification that Rita and Pierre also talked about earlier, separating objects of individual species, or if that's not possible, individual genera or families. And as a starting point for this final step, the fine-grained classification, we are using images that are already publicly available. As Pierre was saying, for the plants, there is a lot of already expert-reviewed images of insects and moths in particular available through GBIF or other platforms, and this data can be used to train a fine-grained classifier for moth classification. Typically, those images would not be from these particular traps, and this introduces some benefits and some challenges. I would say that one of the benefits is that the model trained on this broader and more varied data set will be able to generalize better and maybe even detect and classify insects that are flying or that are sitting in unusual postures. But it also involves the challenge that the domain, the type of images that the traps generate are different, and therefore may not be as recognizable as the images from these public repositories like GBIF. Yeah, next slide, please. So as to alleviate some of these challenges, and actually this goes both for the plant module and for the insect module, we are hoping to involve species experts from each of the partner countries in a process of adding additional data to these public repositories. And this goes to some extent also for the localization, which I think now is working quite well for the AMI traps, or these insect camera traps. But for the plant module, there will be some work ahead as well, both within the pilot and as part of other projects, to improve localization of objects within these broader images, but certainly also when it comes to classification, that even if there are a lot of images already available from public repositories, certain species will be rare. Like Rita talked about, the long-tail distribution of these data sets is very pronounced also for insects, and therefore the traps allow us an opportunity to enrich these data sets, both with more images of rare taxa, but also with images that have the characteristics of these insect camera traps. So the models will be better able to classify insect species that get attracted to these traps. And we're using this annotation tool in Label Studio. There are many annotation tools that allow users to interact with images from monitoring stations or public repositories. Label Studio is one of maybe the more sophisticated tools that will also allow this feedback loop between model and human, or computer and human, if you like, so that that the model can already propose IDs, both of for the localization and the classification task, and the expert will only have to correct the model when it's wrong. And in this way, we make the whole image review process as efficient as possible. And next slide, please. Okay, so I'm ending with a few messages out of this work done in the pilot schemes. I think we're still in the early days of this, but we are certainly learning a lot from implementing these image-based approaches at large spatial scales across both many biogeographical regions where the biological data looks different, but also where the infrastructure and expertise and familiarity with image-based monitoring approaches are different. And this helps us learn a lot about their strengths and weaknesses and what opportunities lie ahead for their large-scale implementation, also for actual management purposes. A key aspect of the Biodiversa Plus pilots is that it's implementing technology that has already been pilot tested. So in terms of both the hardware and the image pipelines, image analysis pipelines, we've had something to build on, but these early stages of testing cannot really compare to the spatial and just this broad scale of implementing Biodiversa Plus pilots. So this is an important step in the implementation of these image-based approaches at the broad scale of the Biodiversa Plus partnership. I think at the moment, what we're seeing is that there are challenges both in terms of the equipment, the data management, and the data synthesis, but that it is realistic that we can have like end-to-end image-based monitoring where the data coming from the sensors will be automatically transferred to the data analysis pipelines and can lead to actionable knowledge that are available at a much shorter interval from the time when the observations were made and when the actions need to be taken. So I think that's a key and very exciting aspect of the Biodiversa Plus pilots. Yeah, I hope there might be a few questions. Otherwise, yeah, we've got lots of people involved with this. Some from the Mambo project, but also many others that are working around the planet to help us implement these tools. So thank you to all of them and to you.
Thank you very much, Togo, for enlightening us on what's going on in the monitoring pilots of the partnership in relation to image-based monitoring. We have still a few minutes left for questions. So if there's any questions to Togo, you are welcome to raise your hand or to write in the chat. I see Pierre, you have a question, please.
Yes, thank you. Thank you very much, Doc, for this great presentation. Can you let us know if you think that the cost of this sensor and the implementation of such methods, you feel that can be a limitation, or is something that should be reduced according to the progress and or the support of national agency, for example?
Yeah, that's a very good question, Pierre. I guess I'm not in a position to identify what the limitations are for individual partners or institutions, but clearly what's generally the the case with sensor-based monitoring is that the investment costs are big compared to pen and paper, if you like, but that the downstream cost savings on manual labor may outweigh that, or the richer data sets that come from these approaches may also outweigh that. But it is true that both of these sensor types that I described in this program are quite costly, and this is, I guess, both a challenge for the implementation phase, but it may also be a challenge if you're leaving some of this equipment in remote locations and you're worried about theft or misuse of the equipment. So I think in the Mambo project we spent some time on cost benefit analysis. I think that may also be relevant in Biodiversa Plus to have more emphasis on and to really cost in all of the associated activities that would be involved with this approach compared to alternatives.
Thank you, Doug.
Thank you both. Any additional questions for Togo before we wrap up? If not, I think we are at the end of this small webinar. So I will just give a very few final remarks as a non-expert in this field, but I can see that biodiversity monitoring is now covering about 1/3 of the budget of our partnership. So it's really a lot. So it's certainly a field with a lot of attention. And I think also, as we have seen today, that these novel approaches and technologies is in constant development, including image-based approaches. And it's certainly amazing what is possible. And that also that there are, of course, challenges. For example, that we need continuation of long-term data series, which are crucial. And I think, as David Roy touched upon in his presentation, that the need of kind of integration and complementarity, so we are not starting over from scratch. So it was our hope that that this webinar has contributed a little bit to highlight some of the challenges and opportunities, and there will be more events in the partnership on novel technologies like this, and we also hope that we have built bridge between researchers and experts, and on one hand, and persons in agencies, ministries, and others on the other, which are part of implementation at national, local, and I mean all geographical levels. So, thank you very much to the presenters, to David Roy, Pierre Bonnet, to Rita Pucci, and to Thomas Høje for presenting today. That has been really fruitful and amazing what you have informed us. And thank you to Cecile for her continuously support with this work package of the partnership. That's really amazing what you're doing, Cecile. And also to Hilde Eggermont for setting the scene in the start. So thank you to all also for joining this webinar, and have a good lunch and have a nice afternoon. Thank you very much.
Thank you.
Thank you too. Thanks to all of you. Bye-bye.